Welcome back, KSTS scientists. I'm so excited to start our unit on genetics today with a brief history of this very important molecule in front of me, which is, of course, deoxyribonucleic acid, or DNA. So today I'm going to go over a few really important landmarks in the history of the discovery of DNA. So I want to take you back to a time when we really didn't know a lot about inheritance and genes and how things are passed on from parents to offspring, and talk about how we got from that point to having a structure of DNA. So the first really important event was when Charles Darwin published On the Origin of Species by Means of Natural Selection. Now, of course, Darwin's book was focused on the concept of evolution, not on genes, but in describing how evolution happens, Darwin talked about inheritable traits, right? Traits that can be passed down from parents to offspring. And of course, Darwin was talking about this in the sense that organisms that have favorable traits will pass them on to their offspring, whereas organisms that have unfavorable traits will not survive or will have fewer offspring. But this sort of first put out there this idea that traits are passed on from parents to kids and that there must be some mechanism through which that happens. A little later on in the 1800s, an Austrian monk named Gregor Mendel, who we'll be talking a lot more about, was busy playing with some pea plants in his garden, and he discovered some really important genetic principles without actually knowing that DNA was behind many of them. And we're going to learn all about Mendelian genetics later on this year. But again, Mendel talked about this idea of this invisible factor that was responsible for visible traits. And he called these invisible factors genes. And although Mendel discovered many of the important principles of how these factors are passed on, he did not know exactly what was this genetic material that was being passed from parent to offspring. All right, a little later in the 1860s, Friedrich Miescher identifies nuclein, or nucleic acid, in the nucleus of white blood cells. So he was actually trying to isolate proteins from inside the nucleus of white blood cells, and the way he did this was by taking bandages that were filled with pus from hospitals and getting the pus off the bandages, because there's a whole lot of white blood cells and pus, and then isolating the nucleus and what's inside of them. And instead, what surprised him was he found this molecule that had a lot of phosphorus in it, which is there's not a lot of phosphorus in proteins. And so because it was associated with the nucleus, he named it nuclein. And of course, today we know these as nucleic acids, which include DNA and RNA. So as the search for these invisible factors or inheritable factors continues, scientists really aren't sure which category of molecule inside our body is the genetic material. Most scientists were putting their money on proteins because we already knew that proteins came in a variety of shapes and sizes, and they seemed very, very complex and seemed like the natural molecule to choose for what might encode our genetic information. But a few key experiments helped prove that it was not proteins, but was nucleic acids, and in fact DNA, that holds our genetic material. And one of them was this British scientist Frederick Griffith's experiment in the 1920s, where he studied a process called transformation in bacteria, which is something we're going to talk more about later, but today we're going to talk about it in terms of why it was important for DNA. So basically, he knew of these two strains of pneumococcus bacteria, a rough strain that had these proteins on the outside that made it look rough, which was non-virulent, meaning non-deadly. And there was a smooth strain that lacked those proteins that was virulent. And when he injected the non-virulent strain into a mouse, the mouse lives. But when he injected this virulent, this deadly strain into a mouse, the mouse died. What was really cool about Griffith's experiment is that he then tried a couple combinations. He took the smooth strain and he killed the bacteria with heat. So he subjected them to intense heat, and then he put it into the mouse, and the mouse lived, suggesting that the heat was able to kill the bacteria. But then he took some of this dead, smooth bacteria, this deadly bacteria, but it was dead, and he mixed it with live, non-virulent bacteria, and the mouse died. So what this suggests is that the bacteria, the live bacteria, was able to pick up something from the environment that came from this smooth bacteria and was transformed, was changed by it into becoming a more virulent or deadly strain. So we now know that bacteria can do this, that they can actually like pick up pieces of genetic material from their environment. So imagine if you could just go around and like pick up pieces of cool looking DNA and make them a part of your genome. But bacteria can do this. But one of the reasons that this was really significant in terms of the search for the genetic material is whatever was causing this transformation was not destroyed by the heat. And it was well known at this time that proteins become denatured or fall apart when they're subjected to intense heat. So whatever this molecule was that was transforming or changing the bacteria to make them become more deadly, it was something that was able to survive or withstand a lot more heat than protein could. So then a scientist named Oswald Avery decided to follow up on Griffith's experiment and really prove that it was the DNA that was causing this transformation. So what he did is he took that mixture of smooth strain bacteria that had been heat killed. So the bacteria are dead, but whatever this genetic material is inside of them, we think that it's still intact because remember, it's able to transform those other bacteria. So he takes this heat-killed bacteria and he subjects it to different types of enzymes. Enzymes are little molecules that break down other molecules. So for example, enzymes called DNases, they eat up or break down DNA. Enzymes called RNases break apart RNA. Lipases break apart lipids or fats. Proteases break apart proteins. And then I'm not sure what is meant by this other category. And then after subjecting the heat-killed bacteria to these different enzymes, then he mixed them with the live R strain bacteria. And what do you know? When the proteins had been destroyed, the bacteria were still transformed and the mouse still died. When the lipids, when the fats were destroyed, the bacteria still transformed and the mouse died. When the RNA was eaten up and destroyed, the bacteria still transformed and the mouse died. But when the DNA was eaten up and destroyed, the bacteria were not able to transform and the mouse lived, which suggests that DNA is what was transforming, what was changing these bacteria, which was really one of the first, most definitive studies to suggest that DNA is the genetic material. This experiment was really not recognized for its importance. In fact, many scientists have said Avery is the scientist most deserving of a Nobel Prize who never got one. It wasn't till much, much later that this work was sort of appreciated for how definitively it shows that DNA. So also in the 1950s, a Frenchman named Erwin Chargaff was studying what are called nitrogenous bases inside of nucleic acids like DNA. They're called nitrogenous bases because they have nitrogen in them. And there's 4 of them, and we're going to learn about them. Their names are adenine, thymine, guanine, and cytosine. Thymine and cytosine belong to a class of molecules called pyrimidines, and adenine and guanine belong to a class of molecules called purines. So what Chargaff figured out is that, first off, the relative amount of these 4 nitrogenous bases is different inside of different living things, in a human versus a monkey versus a plant versus a mouse, which suggests that maybe it's something about these bases that makes the human a human and the monkey a monkey and the plant a plant. But maybe even more important than that, he discovered that there was a relationship between the amounts of these things. So the total amount of purines seemed to equal the amount of pyrimidines, and specifically the amount of adenine, or A bases, was proportional to the amount of thymine, and the guanine was proportional to the amount of cytosine. And these are really going to help develop what are called the base pair rules inside of DNA that we're going to be learning about later on. And then, of course, in 1953, Watson and Crick published The Structure of DNA. We are going to be watching a film in class that talks about how they put together the pieces of the puzzle in order to come up with the structure of DNA. One piece of the puzzle that they used was this X-ray photograph of DNA that was taken by a scientist, Dr. Rosalind Franklin. And it's sort of established that they really did not give her appropriate credit for her work and her role in the creation of their model, and we'll see that play out a little bit more in the films. All right, so here it is, the code of life, DNA. Let's go over some of the things that you need to know about it. So DNA is a double helix, right? It looks like a twisted staircase or ladder, like this, double helix. Okay, the outside of the helix, okay, the outside of the ladder is what we call the sugar phosphate backbone of the molecule. And that's because it's made of two things. You guessed it, sugar and phosphate. So here are the sugars, sugar, 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 and here are the phosphates, phosphate, phosphate, phosphate. Now the sugar is a sugar called deoxyribose. So obviously part of the name of DNA. And then in the center of the molecule, the rungs of the ladder, the steps of the twisted staircase, are those nitrogenous bases that Chargaff studied. So these are the nitrogenous bases. And again, there are four of them: adenine, or A, guanine, which we abbreviate with a G, cytosine, and thymine. And we now know that adenine will only pair or go across from thymine, and guanine will only pair or go across from cytosine. And so if you know which bases are on one side of a DNA molecule, you can tell which bases are on the other side. For example, if one side of the DNA reads A, T, G, C, A, I know the other side must be T, A, C, G, T, because A only goes with T, T with A, G with C, C with G, and so on. And it's really the order of these nitrogenous bases inside of DNA that makes us who we are. So this is the code of life. These are your genes, are these letters inside of DNA, and you may wonder how does the order of these letters translate into making you, and that's part of what we're going to try to uncover during this unit. So that's it for now. Lots more to come. In the meantime, if you're interested in some of the more recent landmarks in DNA, I'm going to post a link to a website that describes some of those. Thanks for watching. Yeah.
Can they see me?
Yeah.
They can see. Oh, I can't see. Oh.
I'll edit this out.
Hey, good luck. You're gonna need it.